Welcome to our series, Microorganisms Through a Different Lens. It's a part of the Master of Beautiful Achievements series, a place where I, Alexander, investigate systemic sustainable solutions using nature as our inspiration. The Microbes Through a Different Lens is hosted by Mark van Bemmel and Alexander Prinsen. And each month we invite a guest in the field of biology, microbiology and biochemistry for an explorative dialogue on how we look at our natural world to discover how this impacts how we look at life sciences, biotechnology, and environmental sciences. And in today's episode, we'll investigate what is so unique about the Amammox bacteria and its impact of our understanding of the microbial nitrogen and methane cycles. And I think it's even more interesting the possible applications this will provide for health, the environment, and I assume much more. But before we start off, let's introduce ourselves. My name is Alexander Prinsen, and with my company Scope Matters, we help businesses bridge ecological thinking with economic doing so we can help accelerate their innovation capacity. We apply natural sciences and systems thinking to ensure circularity is the only logical outcome. And now the word to you, Mark.
Yes, I'm Mark van Bemmel. I'm a founder and CEO of Orvion in the Netherlands. Orvion is a biotech company which focuses on the development of molecular tools to analyze bacteria mainly, but also other microorganisms in water and other environmental things like soil and air. And yeah, we have our own laboratory here in Stolwijk near Gouda, the Netherlands. And we work a lot in this field all over Europe, but yeah. I'm very interested to hear the experience of Mike. We have a lot of experience, of course, with the degradation of all kinds of contaminants, and also looking into sustainability in these water and environmental sectors. So that's our interest.
And now, Mike, introduce yourself.
Hello, my name is Mike Jetten. I'm currently professor of microbiology at Radboud University in Nijmegen, the Netherlands. And a long time ago, maybe I was even younger than our youngest participant today, I got fascinated by the world of microbes during my biology course at high school. I was fascinated by the enormous diversity and metabolic possibilities of microbes. So when I had to look for a study, I tried to combine the best of biology and chemistry, and I studied molecular life sciences at Wageningen University, and I was able to continue that in a PhD on anaerobic microbiology. Then I got familiar with the anaerobes, especially the archaea that make methane. Then I was very fortunate to have an interlude in the United States, switching from the slow-growing, tedious anaerobes to fast-growing industrial workhorses making glutamic acid. I worked on corynebacteria and trying to metabolically engineer them. Very fascinating time at the Massachusetts Institute of Technology, but also glad that in 1994 we could return to the Netherlands, and I joined the Delft University of Technology that at that moment was busy with a very peculiar and for many thought impossible process of anaerobic ammonium oxidation. So I had the pleasure to work with Gijs Kuhn and Mark van Loosdrecht for 7 years at Delft University and the nitrogen cycle and Anamox. And in 2000 I had the opportunity to join the Radboud University to set up my own group in microbiology. For the past 20 years we have been working on digging into the mysteries of the Anamox bacteria. How do they work? What can they do? How can we apply them? And I was able to go back to my methane cycle microorganisms, because we thought if it is possible to convert ammonium without oxygen, then it surely must be possible to convert methane without oxygen. We can see how these discuss how these processes have parallels and differences.
Thank you, Mike. That gives me and Mark a lot of hooks to have to fill up this 1 hour of conversation. I think for those who are listening, Let's start off with a small explanation of what the difficult word of anammox means, and what kind of organisms these are, and what kind of role do they have to play in the web of life.
Yeah, Anammox stands for anaerobic ammonium oxidation. And as many of you might know, we have a lot of mouths to feed on our planet, and to grow enough crops, we can use artificial fertilizers, and about half is natural fertilizers, half at the moment is artificial fertilizers. So a lot of the ammonium that is made is applied on fields, but not everything ends up in crops. And the ammonium that is not beneficial to the plants needs to be removed, because it has a lot of detrimental effects on the ecosystem. Mm-hmm. Now, already maybe even already in the 19th centuries, the early microbes, the Russian microbiologist Vinogradsky isolated two microbes that could convert ammonium with oxygen to nitrite and to nitrate. And so the people thought, okay, apparently ammonium needs oxygen to be converted by microbes. At a certain moment in time, people have been looking for microbes that could convert ammonium without oxygen. Many people tried to find them, but never really succeeded. If you do the calculations, if you do the thermodynamic calculations, if you see that ammonium with nitrite or nitrate, there is enough Gibbs free energy in this reaction that microorganisms should be able to live on it. And, microbes have been on this planet, and they use all chemical reactions where there is a liberation of energy. So for us, it was very clear these organisms must exist. Luckily, Arnold Mulder, he was chief wastewater engineer at the Gisborgades company in Delft, and he was operating. So this factory produces a lot of yeast for the baker and brewery industries, and they produce a lot of penicillin, so antibiotic industries, and they have a lot of wastewater that they wanted to treat anaerobically. In that wastewater, there was a lot of sulfate. If you treat anaerobic wastewater with a lot of sulfate, then you inadvertently produce hydrogen sulfide, very smelly. So Arnold got a lot of complaints from the citizens of Delft that his wastewater treatment plant was producing too much odorous gases. So being a pragmatic wastewater engineer, what he did, he bought a lot of calcium nitrate and put that into his anaerobic system, where he could suppress the sulfate reducers, making hydrogen sulfide. And without knowing, he had created pretty good conditions for the Anamox bacteria to proliferate. So about half a year after he had dumped in the calcium nitrate, he saw that ammonium was disappearing from his anaerobic wastewater treatment system. Eureka! You remember the theoretical publications that this should be possible. So then he contacted Gijs Kunnen at the Delft University of Technology to investigate the biology behind this process. It was also about the time that I joined the TU Delft, and we could really start looking into, is this really a biological process? Is this really bacteria that are converting the ammonia without any oxygen? And yes, to make a long story short, yes, it's truly a biological process, and it took a lot of overcoming resistance to make other scientists believe that these bacteria were real and that they could really do the job of converting ammonium without oxygen.
And we have found them also in nature nowadays.
Yeah, we started out in the engineered systems and anthropogenic, because there is a lot of ammonium and often a lack of oxygen. And in collaboration with our ecology colleagues, and especially with those of the Max Planck Institute in Bremen, we thought, well, if you think about ecosystems on Earth where there is a limitation of oxygen, then you think on oxygen minimum zones, you take on swamps, you take ditches, everywhere where there is a good mineralization of biomass and a lack of oxygen. And indeed, nowadays we can find these organisms in all of these ecosystems. But before we could detect them in these ecosystems, you first have to know who they are, what their genetic material is, because so it's not that easy to find microbes. But once you know their genetic makeup, then the diagnostics become so much easier. Yeah.
And that turned out to be a worldwide revolution.
At least within the microbiology world.
You're too modest, Mike. Yes, of course.
That's what I understood from Mark. You told me that the innovation Mike is working on, the discovery of Mike has created quite a ripple in microbiology and wastewater and beyond.
So it was also because these organisms are very special. They defy many of the dogmas how microbes should look like, how they should act.
And in what way? Can you just give us some examples?
Normally people think of bacteria as bad guys, of course, but the majority of the bacteria are good guys and girls. They are beneficial for our health, beneficial for our environment. Many people think of bacteria as a simple sack of DNA and proteins. But the Anamox bacteria, they have a very complex cellular makeup. These are bacteria that have organelles. Eukaryotic cells, we are all cells, they have different organelles. Mitochondria, endoplasmatics, reticulum, Golgi apparatus. And this bacterium also has an organelle, which is very, very unusual. And it turns out it needs this organelle to contain all its enzymes that carry out very reactive reactions. So in converting ammonium to dinitrogen gas, that's what they ultimately do, they produce very volatile and reactive intermediates like the rocket fuel hydrazine. And to protect itself, hydrazine is a toxic compound, so to protect the remainder of the cell, they have compartmentalized this reaction into a bacterial organelle. And that's very unusual.
So it's really a survivor.
Is that also the reason, Mike, why they grow so slowly?
Depends on what you call slowly.
Several days to multiply, I would say.
Yeah.
Or?
No, I would say if you look into the environment, then many, if not most, microorganisms are nutrient limited. And maybe in a wastewater or an industrial setting, that's where they have plenty of food to proliferate. But generally, most microorganisms live under starvation and have very slow growth rates. Yeah. So I would say for the natural settings, it's quite adapted. There is not enough food to grow fast. So they have not invested in fast growth. They have invested in being very effective and very efficient in taking up the ammonium. And they probably can do so much more effectively than other organisms and be in this way competitive. They just suck in the ammonium before others can do, and then they have the food for themselves.
Smart guys.
Yeah.
Smart guys. And I was doing research, Mike, and I also found the other sides, which is the Amammox. Oh man, these difficult words for me.
Yeah. This stands for complete ammonium oxidation.
And are they the sort of the same family, or are they completely different organisms?
Within the world of microorganisms, there is a huge diversity.
Mm-hmm.
Maybe even this week had a discussion with one of my students on how different organisms are. So maybe people all think that all bacteria are closely related.
No, but
they aren't. So there are
No.
huge phylogenetic differences. So no, the Amammox bacteria and Anamox bacteria have very little in common, except that they convert nitrogen compounds.
And they don't live. Do they live together in the same environment, or are they completely?
They can. Which was a surprise to us, but they can. Maybe. Yeah, to go back to the Amammox bacteria, that also there. So I said Sergei Vinogradsky isolated ammonium oxidizers that used oxygen to convert ammonium into nitrite and then nitrite into nitrate. And also here, some colleagues of us produced a theoretical paper that said there is thermodynamically, chemically, no reason to have these reactions separated. There should be organisms in nature that can do this in one go, that can catalyze both reactions. And probably these organisms have specialized under low ammonium, low oxygen, and low growth conditions. Okay. We are maybe not very actively looking for these organisms, but at a certain moment, I had one of my big European projects, and Maartje van Kessel, she was a PhD candidate on that project. We had a aquarium system in the basement of our faculty, and in the wastewater treatment systems of this aquarium system, there was we thought there were some very interesting Anamox bacteria. And I asked Maartje, could you please isolate these interesting, potentially new Anamox bacteria? Okay, she went out, she set up a bioreactor, inoculated this with biomass from the biofilters of the waste, it's called the recirculation system, and started to feed them ammonium, nitrate, nitrite. And then after a good year of enriching, she saw a combination of what we thought were Anamox bacteria and nitrite oxidizing bacteria, Nitrospira bacteria. Very curious, because normally these are competitors for each other, they compete for nitrite. But nowadays, and Mark will smile if I say this, nowadays the fastest way to find out what an organism can do is sequence its genome. So we isolated the DNA, sequenced the genome of the whole community, and then surprise, surprise, this Nitrospira bacterium contained the diagnostic genes for aerobic ammonium oxidation. So it contained the ammonium monooxygenases.
Oh, wow.
And so that, it has both the aerobic ammonium oxidation pathway, it has also the nitrite oxidation pathway. Yes, this must be an organism that can do both.
Oh, right.
And then, of course, we did a lot of experiments with substrates, labeled substrates, more sequencing, more microscopy. And we could finally confirm that, yes, indeed, also these complete ammonium oxidizers do exist. And also, this has also now taken off. Once you identify a certain group of organisms capable of catalyzing a certain reaction, and once you know the diagnostic genes, then you can develop PCR primers, you can look in wastewater treatment systems, you can look in paddy soils, you can look in swamps, you can look in marine systems. And once you know the diagnostic genes, you can easily find them.
Right. So that also poses the question, Mike, is there still a lot that we do not know? I mean, we have a lot of sequences that we do not identify in the end. So
yes, in general, I think we only know a few % of the bacteria, archaea, microorganisms that are out there. Whether it's 1 or 5%, I wouldn't know. Different opinions on the different surveys.
But that's not
and with
only 5.
With the rapid 5 to let's say 5. We know 5% at the upper maximum, I think. Yeah. But with the rapidly advancing sequence technologies and people sequencing whatever sample they can get, I think the sequence space will be filled within a few years. Then maybe we have sequenced it all. what it means is that something completely different. And even I think you do a lot of diagnostics for several different companies, and also in your samples you'll find that 50% of the genes you cannot quantify, qualify, and
or only to a certain extent. So we know it's that or that family with no further genus or species information. Yeah.
I look at it from a different perspective. Yes, probably there is a lot of sequence space we can fill and can find out. I normally try to ask my questions by metabolism, and then I translate Alexander's question: are there still several nitrogen cycle microorganisms that should be out there? And yes, I think so. We would like for example, in the microorganisms, we make a difference between the bacteria and the archaea, and we have seen that there are both bacterial and archaeal ammonium oxidizers. Then the same we can ask: are there also bacterial and archaeal nitrite oxidizers? There are a lot of bacterial nitrite oxidizers, but so far no one has found an archaeon that can do that. Well, there should be one. No one has found them.
But from a thermodynamic perspective, there is enough energy for niches to do something.
Yeah, and the same. Why is there not an archaeal Amammox, or why is there not an archaeon that can do Anamox?
Yeah, I just wrote down here. It's like, I mean, I also do a podcast series in Dutch from the agricultural system, and we had the guy called Piers Flores, who's doing, let's say, soil restoration with, for instance, fungi. And what you're mentioning is that, I mean, plants and nitrate or ammonia are, of course, very closely related. I mean, they need to get the stickstoff, or the nitrogen, some nitrogen intake somewhere. But the way you are explaining it is that I feel like the soil has so much more for us in petto, which is the Dutch word for so much more to share, because what I'm learning from you, Mike, is that the ammonia or the nitrate cycle, the nitrogen cycle, is quite complex, and yet it can provide so much more energy when a bacteria finds its pathway in there.
Yes, the more we study the nitrogen cycle, the more complex it gets. It doesn't get simpler and easier. So in the past 20 years, we have seen so many fantastic discoveries: archaeal ammonium oxidizers, Ammox, Anamox, so many different reactions. We have found them in different places. So yeah, it has been a fascinating time that many of our colleagues have contributed to making, on one hand, the nitrogen cycle more complex. It's not as easy as we thought, and finding all these new microbes that have been on the earth for billions of years already doing these reactions without us knowing what all is possible.
And actually the Anamox bacteria was turned into a very efficient wastewater treatment technology.
Yeah, and that is then the benefit of working at a technical university. The collaboration with a group of Mark van Loosdrecht is in that sense very beneficial, and we complement each other. And I think in the end of the 90s, it was clear that we had discovered what Anamox was, and that there also was a potential within the wastewater industry. And we always had been from the anaerobic perspective in good contact with the Park company in Balk, Friesland. And going from a scientific discovery to a company and to an application is a whole different ball game.
Yes.
Took quite some convincing for Park to take a license. Having a license is okay, but then you also need a pilot plant. I was then in the process of composing, writing a big EU grant, bringing several European groups together to study the Anamox and nitrogen cycle in more detail. And as what is now, and also as was then, it is good to have bigger and smaller companies involved. And then we discussed with Mike and say, okay, the EU doesn't know how big a pilot plant needs to be, or to how small or how big. So let's put in a few 100,000 liters and see what we can do. So we skipped the 1,000 liter scale and really went to the 100,000 liter scale. It was a big gamble, maybe not from the biology, but from the engineering perspective. And we ran into a lot of pragmatic problems, that suddenly we had a winter of minus 20 degrees in the Netherlands, which never happens. But then the pipe froze, which delayed the process. But ultimately, it was a great collaboration between the Waterward, Hollandse Delta, and Rotterdam Park and the TU Delft to get that plant running. And yeah, once you have the first one running, you have 100,000s of liters of Anamox bacteria, and then you can start. So the startup of the first one was a real challenge. It took us more than 2.5 years to get that up and running. But once you have a reactor with 200,000 liters of Anamox bacteria, then seeding the next one is not such a big challenge anymore.
That's much easier, yes. And what I found also on the internet is that these Ammonox bacteria are very acid tolerant. Is that correct?
Then you have read other stories than I have.
I found an article that methane bioconversion to methanol by acid tolerant ammonia oxidizing bacteria.
Very good that you found last week, isn't it? Or 2 weeks ago.
It could be.
That's very recent. I've only seen the abstract. I've not read the paper.
Okay.
Anamox bacteria have many, many interesting features, and I'm sure some are more acid tolerant than others, but real acidic Anamox bacteria I've not yet come across. I'm sure they will exist, but we haven't found them. What we and others do have found is very, very acid tolerant ammonium oxidizers for example, acidic volcanic systems in Yellowstone. And not surprisingly for the Dutch in, what is it called? Biofilters for when you have pig farmers, they have a lot of pigs with a lot of stables, and the air of the stables is full of ammonia, and this air has to be washed and cleaned before you can emit it. And so you have these acid gas washers, how they called biofilters. These are very acidic, and they contain very peculiar acid tolerant ammonium oxidizers.
Oh, interesting.
Yeah.
But that is also the basic of technology, or that is an unintended consequence that actually they found a niche in these filters.
Yeah, if you have air with a lot of ammonium and you pass it through a water filter, it gets oxidized. Nitrification produces a lot of protons.
Yeah.
And protons by default if the water is not buffered, they make it very acidic. And what normally happens is the nitrification stops if it gets below 6.5, But these acidic ammonium oxidizers, they have specialized that they can tolerate these very acidic conditions. Yeah.
Oh wow! And it is interesting. I mean, this is a good nature-inspired technology just to do scrubbing from a bacterial perspective and not from a chemical perspective.
Yeah.
Oh wow!
Mike, in Holland we have too much of this ammonia.
We are in a crisis, aren't we?
Yes.
Corona, a nitrogen crisis.
They call it a nitrogen crisis. Yes, maybe that's a bit strange for our guests from the USA, but in Holland we have too much of this ammonia, and it deposits in nature reserves and influences the species composition, mainly for rare plants, I would say. But in a way, Mike, do you think the microbes could help in solving this issue in some or another way?
My scientific opinion or my political opinion?
Yeah, whatever. I don't care.
My scientific opinion is that yes, bacteria could help, but they must not help us to legitimate maybe the way we treat our planet.
True.
Of course, we have now a very big political discussion whether we, in this very, very small country with 17, 18 billion people, should also house millions of animals. So I think we should house less animals. And we have very big countries on our planet where there is maybe a better balance between people and animals and nitrogen emissions. So first of all, the solution is not only in the microbes, also in producing less ammonia. That is the fastest way to get rid of our crisis. And yes, in niches, bacteria can help.
Right.
And yes, we do that already in wastewater. We do that already for 50, 60 years in wastewater. The bacteria do help to convert the ammonia. And then you can ask, what is the wisest? Anamox does it very sustainably and removes the nitrogen gas. Maybe there are also processes where you could recover part of the nitrogen and use it as fertilizer and not convert it.
I mean, we talked with Willy Verstraete last December on that topic.
Yeah.
Yes, he wants to build proteins and feed the world with it. That's sort of his vision.
And using sustainable nitrogen instead of the chemical one.
Well, before we can use the nitrogen from a wastewater into a feedstock product, I think before anyone of us would like to do that, you need safety and food safety guidelines. Yeah. So I think a lot of technology and science has to be done before we are at that stage. Using it fertilizer might be a little bit easier. If you can put cow dung on a field, then you certainly can also use surplus sludge.
Right. Yes.
Yeah.
Mark?
Using bacteria for single cell protein, that idea has also been around for 50 years. And then maybe not only for ammonium, but also for methane eating bacteria, but it never has really taken off.
No.
What are the bottlenecks in that case?
I haven't read about it recently, but these methanotrophs, they have too much urea or urealytic content, so that the quality of the protein for feedstock is not good enough. The cows don't like it.
That's something I was also considering. If you have in Holland, we have a lot of this manure, obviously.
Yeah.
And, we did some work on it in our lab to convert it to make part of the organic content of the manure to methane. Obviously, that is it works to some extent. You can get some methane out of it, and then you you have some energy. But also you produce pretty high ammonia concentrations during the fermentation process. And if you could also convert part of that ammonia, or at least make it a bit less, and it could help by creating both methane and also reduce the amount of ammonia that you release back into nature. If you understand what I mean, so.
Not entirely, but the conversion of organic matter, then unfortunately, organic matter biomass is 50% protein, and protein has a certain stoichiometry of nitrogen and carbon that you can't change too much. So if you convert biomass into methane, CO2, then by default you also produce about 15% of the biomass is nitrogen.
Exactly.
And there is not much you can do about that
The
You
free
will.
ammonia that you produce, you could maybe reduce that.
I didn't hear.
The free ammonia that you produce during the fermentation process, maybe part of that can be removed by Anamox or other kind of bacteria.
Yeah, certainly in a Sidestream that's possible. And
Yeah.
that's already what is happening in many wastewater treatment systems, where the Sidestream ammonium, whether it's from reject water or from leachate, that is certainly possible.
Yeah.
And as you have connected the nitrogen and methane cycle so nicely, that that's what we also have been investigating in the past 4 or 5 years in many wastewater treatment systems. But you have anaerobic digesters, so they produce this nitrogen concentrated stream. Therefore, you can very well apply the Anamox process sustainably. And we were also thinking, so some 10 years ago, we discovered other bacteria and archaea that can use the methane. So what we in the Netherlands now do is we have a lot of sewers that bring the waters to the wastewater treatment plants. There we convert the ammonium to nitrate. Then we buy a lot of methanol to convert the nitrate into dinitrogen gas, and this should be possible in different ways. So one solution is the Anamox, so that the Sidestream nitrogen is removed. And we thought, could we also use the methane that is generated on site and which is sometimes even saturated in the waters, to use that to polish the nitrate? And yeah, the biology is available, no problem. We have the bacteria and the archaea that can do the job. But the economics are not yet. We have done some calculations, market analysis, but the economics are not yet there.
How is this compared to the technology of Lanzatech?
Then you have to summarize what you.
Yeah, Lanzatech used to be a New Zealand company, now moved to the USA, and I know they're using gas fermentation, and at least for the carbon dioxide, carbon monoxide, and I also know, also read they are using methane in gas fermentation to produce among others, biofuel. But is gas fermentation a limitation at the moment in technological perspective?
No. I know a lot of the syngas works. No, the gas. It is not the gas that is the challenge. Within the Dutch wastewater treatment system, there is a levy on the nitrate and ammonium that you discharge. So the better the quality, the less levy you pay. At the moment, there is absolutely no regulations or standards for methane emissions.
Right.
So, what we if there would be CO2 or nitrogen, but also methane standards, then we could measure, monitor, and levy emissions, and then people would have an incentive to be as sustainable as possible. So, and we can foresee in the future, whether it's in 5, 10, or 20 years, for surely there will not only be a CO2 tax, but there will also we will have a methane tax.
But although methane is a good energy provider, I mean, if you oxidize that, then you have your energy source. But then you have
that's already happening in many wastewater treatment systems. They try to purify the biogas, scrub it, have good methane, and put their generators to work. That's for sure a good idea, but there is a lot of diffusive escape of the methane in many systems, but also in wastewater systems, and that's not monitored. I'm not saying it is a huge contribution, but we should try to reduce emissions wherever we can.
Yeah, or put them to use through biology. I mean, biology is smart as hell. I mean, the conversion capacity of biology, or at least microbes, it's so amazing. That's what I've learned from the past series. Every time you never stop amazing what they can do, and if you feast, or if you feast them, or what's the other word? starve and feast them, they turn into they switch new metabolic pathways nobody knew they existed in them.
Yeah.
And that is so amazing that they have the capacity to do so.
Yes, that's why I was fascinated 40 years ago to start my career in microbiology, to investigate the sheer unlimited possibilities of microbes.
Yeah.
Yes. And I found also an article or quote that said marine and ammonia oxidizing archaea and bacteria occupy distinct iron and copper niches. The mineral deposits and these bacteria, these are also connected.
Very good. You should do my literature surveys. I was busy with them before I joined this podcast. This has to do that the archaea and the bacteria use different biochemistries. So the ammonium monooxygenase is a enzyme that contains both iron and copper. And in the respiratory chain of the ammonium oxidizers, there are a few other copper-containing proteins that help them make the living. And what we see in the archaea that it is more iron-only based. So that's why you can imagine iron is a very limiting compound in the ocean. That the archaea will go to the places where they can scavenge the iron, and that the bacteria are maybe a little bit less picky, that they also can go to the places where there is copper. And that in that way they can occupy different niches determined by the copper and the iron availability. And then Anamox for sure needs a lot more iron than the archaea, so that's why you will find Anamox in the deeper oceans.
But if you would have an iron mine, if I would be a mining company and one of my resources is iron, then I would also find a lot of Ammonox bacteria then in the soil.
Yeah, mines are mostly aerobic, so you would have to go deep where there is not much oxygen.
It's interesting. I mean, the alchemist always said you never can turn lead into gold, but now I've learned that there are actually bacteria that can take one electron and put it on another molecule, and then that alchemistry becomes very close. They can tinker around with the molecular structure of minerals, and that is so amazing.
Yeah.
We had from Deltares, we had we had here on the podcast, Mike. We talked about microbial fuel cells.
Jan Gerritsen.
Jan Gerritsen, yes. And I've done also I found also a article or a story about microbial fuel cells and the Ammonox bacteria.
Yeah, that's on our future dream, and we have collaborated with colleagues in Saudi Arabia on this topic. So if you think that Anamox bacteria can use ammonium and nitrite to convert it to dinitrogen gas, then maybe the nitrite could also be replaced by iron oxides or by an electrode. That the Anamox bacteria need something that accepts their electrons. And Dario Shaw has done some tremendous work on this. He has put all his energy and PhD time on this topic, and we managed to get the Anamox bacteria to donate the electrons to an electrode, and in this way you can make electricity from ammonia.
That's super. That's a bit like the story of Peterffy that he told us with his Microbia species.
Yeah.
But are they sort of related then? You think, Mike? This Acidomicrobia and the Anamox, or?
No, not at all.
Not at all. Okay.
But
Interesting, but they could be in a way because they do the first part. Yeah.
Yeah, I've communicated with Peter too. I think the microbia have a completely different metabolism.
Right.
Yeah.
But then again, I mean, I've seen examples of microbial fuel cells that they can power lamps on desk using a plant, and there are now various startups who are using the electron abundance in soil to power small LEDs. But Jan Gerritsen mentioned, and he was quite skeptical. He did his analysis and he said microbial fuel cells and the energy harvesting capacity is still not, let's say, that big enough to do real cool innovations with them. Now it's just street lights and desk lamps. But do you see a bigger, brighter future with these Ammonox?
We are at the beginning to. Yes, we have discovered the biology, and the same as with the implementation in wastewater treatment systems, it takes 5, 10, 20 years to develop the technology to develop enough efficient harvesting electrodes. And I've not done the calculations, but we see amazing developments within 5 to 10 years, so I wouldn't write it off immediately.
Oh, that's good.
What we think now is a fantasy is a reality sooner than we anticipate.
Sooner than you think, yes.
Yeah.
And there's really a lot of stuff happening in this microbial field, fuel
field, yeah.
cells, yeah.
I mean, it's sort of like the holy grail, because then you sort of the electrons are there on other molecules, and if you can rip them off and put them to use, then you have a sort of like a cheap conversion. Yeah. In that case.
At a very low temperature, at relatively dilute solutions. Yeah.
Yes.
They will never generate gigatons of energy, gigajoules of energy, but.
No, but
it could be very localized. Yeah.
Yeah, that's why I mean, only human kind of industry needs a lot of electrons to convert something that doesn't want to move into a different state only when you put a lot of energy in it.
Yeah.
But that's characteristic.
Yeah, I would like to ask the other people that are joining if they have questions for Mike, and then I think it's good if you ask them right now. What do you think, Alexander?
Yeah, sure. Go ahead. This is
Tyler or Daniel or Fede or Paul, if you have questions, then.
Well, I guess I have a question that goes back to my original one, and just trying to understand a little bit more of, again, Anamox is a critical part now of wastewater treatment. A lot more of these systems are being installed out there, and is there really a difference in just the bacterial populations or the different types of Anamox out there, and are there ones that are more beneficial for certain environments than others? Do you see a low temperature Anamox that's potentially going to have the benefits that these high temperature streams have in the future?
Yes, for sure. There is a high diversity of different Anamox bacteria. Do we always exactly know what their niche is? Unfortunately, no. But we and others try to find out. If you look at the Anamox bacteria in the sea, and we have been to Greenland and to Antarctica, in sea ice you can find Anamox bacteria living at -2 degrees. So for sure there are Anamox bacteria that are adapted to cold environments. Maybe they are also adapted to low ammonium concentrations. So we would have to find a niche where we could crank up their capacities. And I believe my Chinese colleague, Gu Bingzhu, has been in the Gobi Desert, and he has found Anamox bacteria in Gobi soil that during daytime can easily get 80 degrees.
Wow.
Yeah. So there is a large diversity, and we haven't harvested that diversity and investigated them. And then probably in wastewater treatment systems, you would like to have Anamox bacteria that can cope with dynamic systems. That might be other ones that can withstand low temperatures or high ammonium concentrations. And I guess the ones that can handle organics and ammonium under dynamic conditions might be most beneficial. But I'm sure that if you build and operate your plant system, that the good ones will come.
Yeah.
Maybe it will take time, but you will get an adapted Anamox community in your wastewater treatment system.
Yeah.
Maybe the question becomes how much patience do you have? How much money does your company have and want to wait?
Yeah. That's what I've always envisioned, but I just didn't know if there were new kind of developments. And again, having low temperature Animox, there's lots of different, it's in a different environment at that point, like you said. So the dynamics are much different in the bacterial populations that it's competing against, whereas the high temperature seems to have some advantage. And as long as you're out selecting these other nitrite oxidizing bacteria, you get that benefit. But on the low temperature side, it just doesn't seem that anyone's making any major steps in that direction yet. So I was just trying to see what the perspective was.
With the lower temperature, I don't think Anamox is the bottleneck. My feeling is that the aerobic ammonium oxidizers are the bottleneck. Not necessarily Anamox.
Yeah, we always know those ammonia oxidizing bacteria tend to be the bottleneck on Sidestream as well. They're a little finicky for sure.
Can you sort of explain, Mike, what happens there with these ammonium oxidizing bacteria? So.
No, I don't know the trick. If I knew the trick, I would have solved it.
You written a paper, yeah.
Yeah.
But, Tyler, do you have a question? Because there's one question that's popping up with me.
So you talked about how copper and iron are often needed in the pathway for these Ammox bacteria. Are there other micronutrients that are required to make that pathway work? Did you hear that?
Could you repeat? I missed the beginning of your question.
So you were saying how copper and iron are needed in the pathway for the Anamox bacteria. Are there other micronutrients required that sometimes get neglected?
Iron is the most important one, and there might be a couple of other trace elements. Nickel, I can think of, copper, that might be important. And it might also be that maybe in wastewater treatment systems, Anamox live in such a nutrient-rich and cofactor-rich environments that they become lazy and depend on others. Why should, if you live in a rich environment, why should you synthesize all the things yourself if you can also get them from your neighbors? And so then they might become lazy and depend on others and lose genomic information that make them dependent.
And that transitions the population from one type of Ammox potentially to another.
Yeah, potentially, yeah.
The question I had, Mike, was, I mean, we know there are some people who want to go off this planet to another planet.
Yeah.
And we know if you do that, you need to bring Earth with you. We will have wastewater treatments because we have human feces that has to go somewhere, or at least the water reuse cycle.
Yeah.
Then you get two questions. One of them is, would there be a chance that from an astrobiology perspective, these Ammonox certainly already exist on certain planets? And the second question would be, can we bring our own Ammonox bacteria with us to do the wastewater recycling?
Yeah, let me first start with the second question. Our Belgium colleagues in Antwerp, Siegfried Vlamings, is doing beautiful work in this respect. It's the Melissa project, making a sustainable Mars mission. I think that's the ultimate goal. And yes, you need to cultivate your own food. You need to recycle all the water. You need to recycle all the nutrients, and for sure, Anamox can help in there and making a sustainable waste system. Sometimes journalists take this another step. can Anamox produce enough rocket fuel to bring us to Mars? I'm not sure. I'm not sure how much the astronaut needs to drink and eat protein bars to make that sustainable. So that's one. And your first question is much more difficult to answer. Is there life on other planets? That's what you're essentially asking.
Yeah, and the reason why I'm asking is that ammonia or nitrogen tends to be abundant on certain places. So it's one of these elements that tend to be there in as a gas or maybe liquefied.
The Anamox doesn't need much. It needs ammonium, a little bit of oxidized product, iron oxides and CO2. That's what it needs. Maybe some water, that would be helpful. If you have gaseous or liquid ammonium, I'm not sure whether that's a habitable planet.
But, and some phosphate and some other, of course, building blocks. But I mean, based on what you shared with us today, that should not be unrealistic to contemplate on.
Yeah, whether all evolutions on other planets follow the exact same path as on Earth, I doubt it. I think that probably much more solutions than we ever can imagine.
Okay, fair deal. But it's just a curiosity, because space travel tends to confine circularity in a very small space, and finding solutions requires to think a little bit further out of the box and make new or strange connections to solve these wicked problems.
And bringing anaerobes to Mars should certainly make sense, because the oxygen is very limited. And the stuff you need for your people if you want to keep them alive there. So.
Maybe Mark, maybe we should get somebody from the Melissa project just to elaborate on that area a little bit further.
Yeah. A few years ago, I was invited to attend a Melissa conference. It was very fascinating. A completely different mindset on the problems that I never had thought about. Indeed, how do you bring 10 or 100,000 liters of water with you on a Mars mission? We don't have the rockets to transport it.
Not yet. They're working hard. The people are working hard on that. Maybe the last note from Paul. Do you have some ads or?
I have a question. I was wondering when do we have the first archaeal Anamox wastewater plant, and what would be the benefits then?
I can only fantasize. First, we need to discover this organism, whether it exists or not. As soon as we find it and have the diagnostics, I'm sure that Mark and you and all the Orvin co-workers will be able to detect it whenever the conditions are right. Does it make a difference for the practice? No, not much, because also wastewater treatment plants, when operated efficiently, have done their job. And it helps to understand how the community in the wastewater treatment system is composed, that you know how to operate it and operate it effectively. But whether an archaeon or a bacteria does the job, no, that doesn't matter that much.
Okay.
Yeah.
As long as it gets the job done.
Yeah.
Okay. I think Mark, should wrap it up. And
so maybe somebody has a question that you still another question that you want to ask to Mike. Maybe I just have one last question for you, Mike. You started with thermodynamics, and you said we knew it should be there, this Ammox, and then it was found, and you did a lot of work on that. And maybe have you ever figured about another thermodynamically favorable possibility that you would like to look into for microbes if they exist? Or
luckily, I have had a very prosperous and long career, so now I can think of other things for the future.
Yes, exactly.
What I said, we have from ammonium with nitrite, we have moved on to ammonium with iron oxides and fuel cells.
Right.
The same thing we have done for methane and nitrate, and at the same time we are also now doing for methane and iron oxides and fuel cells. And if we can wrap that up in the next few years, then I think I've contributed enough to microbiology and leave some room for others to contribute to for the next generation.
There's still a lot to be done, I would say.
Yes.
Yes.
And what would your suggestion be? And then it's a good one to conclude for the next generation. What would be your insight or foresight on how they should approach sciences in the field that you're operating in?
Now, what we now can see is that if you have the genomic information, you can design much better experiments, much better diagnostics, and maybe you don't need the long, tedious enrichment times of years that we had, but that you can, within a few weeks or few months, say we are going in the right direction or we're going not in the right direction, so that you can monitor much closer, and then you can abort missions that are not successful, and then you can start many more lines in parallel than one by one. So
Right.
much better diagnostics.
Yes.
And that requires a much better collaboration between IT, Mark, how do you call them? ICT people?
IT people, yes.
Bioinformatics, yeah.
Yes. Yes.
That's what we, at our department, we have that integrated. So we have people doing experiments, people doing the analysis, talking to each other, making the designs, doing the better diagnostics in a very fast cycling mode.
Yeah, that's really the way forward. I fully agree on that. That's it.
How surprising!
Yeah. That's
So,
yeah.
Mark, shall we wrap up? I wanna thank you, Mike, for the time you took to join us, and
You're welcome.
I want to thank all the participants for their questions, and good for you taking time to be here and from 2 or 3 continents even today. So that is great. This is a monthly session, so there will be a next speaker next month. I think it's already the 22nd of April. The announcement will come next week. If you're interested, just let us know, and then we'll sign up and we do another Teams webinar.
Okay. Have a great Eastern time, and thank you very much.
Thank you very much.